Good evening, everyone. So, today we'll start our session. Today is Tuesday and it is physiology. Today we will study about hemoglobin and its details. Okay, without further delay, we'll start. Are you able to see the screen? Are you able to see? Is it clear? Okay, we'll start. So, physiology and biochemistry, class 2. The topics of today's discussion: illustrate the structure of hemoglobin, discuss the synthesis of hemoglobin, define normal function of hemoglobin, state normal value of different varieties of hemoglobin, explain iron metabolism. So, hemoglobin. Hemoglobin is a conjugated protein in the red cells. It constitutes more than 90% of dry weight of these cells. It transports oxygen from lungs to tissues and carbon dioxide from tissues to lungs. So, the structure of hemoglobin is a combination of heme and globin. Heme is the iron part, globin is a protein part. That is why it is a conjugated protein. Heme is iron, globin is protein. So, heme is a complex molecule. It is made up of tetrapyrrole rings with iron in the center. Okay, so there are tetra is 4. Tetrapyrrole, 4 pyrrole rings with iron in the center. This is heme. This along with globin becomes hemoglobin. Okay. Next, globin. Globin is a protein that consists of 2 pairs of polypeptide chains. Okay, it contains 2 pairs of polypeptide chains. Each amino acid chain is attached to a heme moiety to form a single hemoglobin molecule. So, amino acids are attached at different positions. As we move further, you will understand. Okay, amino acids are attached at different positions to form this peptide chain, and these two pairs of peptide chains together with heme, they join heme, get conjugated and become hemoglobin. Okay, so steps of synthesis of hemoglobin. So the raw materials are iron and amino acids. The raw materials for synthesis of hemoglobin: iron and required amino acids. Okay, so the first, succinyl coenzyme A plus glycine. Glycine is amino acid. In the presence of ALA synthase and pyroxidol phosphate, it becomes alpha amino beta ketodipic acid. This is converted to delta amino levulinic acid, that is ALA, amino levulinic acid. This is converted to porphobilinogen, which is converted to protoporphyrin 9. This becomes heme. Porphohemologen becomes heme. Heme with globin becomes hemoglobin. Okay, so this is the synthesis of hemoglobin. Functions. So, what are the basic functions of hemoglobin? Transportation of oxygen from lungs to tissues by forming oxyhemoglobin, and transportation of carbon dioxide from tissues by forming carbaminohemoglobin. So, transportation of oxygen is most important. More than 95% of oxygen is transported through hemoglobin from lungs to tissues, because hemoglobin has a great affinity for oxygen. Hemoglobin loves oxygen. Okay, so that it gets stuck to oxygen very easily. And one molecule of hemoglobin can carry 4 molecules of oxygen because it is made up of tetrapyrrole ring, as you saw earlier. Okay, so there are 4 iron is in the center. So the tetrapyrrole ring each can carry 1 molecule of oxygen. Okay, and but less than 30% of carbon dioxide is brought back as carbaminohemoglobin from tissues to lungs. The more than 60% of carbon dioxide is brought back as bicarbonate ions or weak acid, carbonic acid or bicarbonate ions. Okay, so when fully saturated, 1 gram of hemoglobin carries 1.24 ml of oxygen. So with when it is when the hemoglobin is exposed to increased pressure of oxygen, 1 gram of hemoglobin can carry up to 1.24 ml of oxygen. So each molecule of hemoglobin will bind to 4 oxygen molecules when exposed to oxygen at increased pressure. Okay, hemoglobin also acts as buffer in maintaining blood pH. Hemoglobin also acts as buffer in maintaining blood pH. Hemoglobin serves to destroy physiologically important nitric oxide molecule. Hemoglobin helps in destroying the physiologically important nitric oxide molecule, and hemoglobin imparts red color to blood. The mammalian blood, the blood from mammals is red in color because of presence of hemoglobin. Okay, next, types of hemoglobin. There are 2 types of hemoglobin: normal, abnormal. So normal hemoglobin, adult hemoglobin, fetal hemoglobin, embryonic hemoglobin. Okay, abnormal hemoglobins are hemoglobin S, hemoglobin C, hemoglobin D, hemoglobin E, and hemoglobin M. These are variations are based on the positioning of amino acid. Okay, so first we will study normal hemoglobin. Adult hemoglobin, it is a major hemoglobin and comprises about 97% of hemoglobin in adult red cells. Okay, it is also called as HbA, adult hemoglobin, hemoglobin A or adult hemoglobin. Okay, next is fetal hemoglobin. It is the major hemoglobin in intrauterine life. Okay, once the baby is born, this fetal hemoglobin gets converted to adult hemoglobin within 8 to 10 days. That is why in the beginning there is physiological hemolytic jaundice in children, okay, in newborn babies. Next, embryonic hemoglobin. This is confined to very early stages of embryonic development. In the stage of embryo, this is present, embryonic hemoglobin. Next, abnormal hemoglobins. There are 5 clinically important abnormal hemoglobins. First one is hemoglobin S, where valine replaces glutamic acid at 6th position. It causes, so what it causes? It causes sickling of red cells on exposure to hypoxia. When there is less oxygen level, it leads to sickling of red cells. HbC, lysine replaces glutamic acid at 6th position. This, what it does, it shortens RBC survival. So the lifespan of RBC is 120 days. This makes the lifespan of RBC short. Hemoglobin E, glutamic acid replaces lysine at 26th position. It produces microcytosis, that is the cells are smaller than normal. Okay, HbM, tyrosine replaces histidine at 63rd position. This also causes problem. Okay, so these are the various types of abnormal hemoglobin. Okay, normal values. Adult males, the normal value is 14 to 18 gram per deciliter of blood. Adult females 12 to 16 grams per deciliter of blood. Newborn babies 16 to 22. See, this is so high. This is fetal hemoglobin. It gets destroyed and it is replaced by adult hemoglobin. Okay, gram per deciliter of blood. 2 to 4 months, 9 to 14 grams per deciliter of blood. 10 years of age, 12 to 10 to 18. Again normal, 12 to 14 grams per deciliter of blood. These are the normal values of hemoglobin. Okay, next we will we will see iron metabolism, because iron is one of the raw materials for processing hemoglobin, and iron is the most important compound, because in hemoglobin iron is the one which has affinity to oxygen. Okay, so sources, rich sources of iron, all non-vegetarian sources, liver, heart, kidney, spleen. Good sources, egg yolk, fish. Vegetarian sources like nuts, figs, dates, spinach, apples and bananas. So most, these are the most important sources of iron. Okay, this should be what do you say, this should be included in the diet. Daily requirement, infants 10 to 15 milligrams, children up to 4 to 10 years 15 milligrams, older children 11 to 16 years 18 milligrams, adult females 18 milligrams, during pregnancy also it is 18 milligrams, females after the age of 50 years 10 milligrams, because after menopause, see, usually iron is lost during menstruation every month, so with every cycle, so that is why there is more requirement in reproductive age. Okay, once there is menopause, there is no regular shedding of endometrium, that is why iron iron requirement also reduces. Okay, so the normal concentration of iron in blood is 75 to 175 micrograms per deciliter. Absorption, how is iron absorbed? This is very important actually. So iron in food, it occurs in ferric state, that is Fe+++. Okay, this is converted, cysteine and ascorbic acid, that is vitamin C converts the iron, ferric iron to ferrous iron in the stomach and duodenum. Okay, in this form it is ready to get absorbed in the stomach and duodenum. So cysteine and ascorbic acid, that is vitamin C, is very important for iron absorption, and vitamin C and also the acidic environment of the stomach, these two are very important, because vitamin C in presence of stomach hydrochloric acid helps in conversion of ferric iron to ferrous iron, ferric form to ferrous form, and ferrous form is the one which is readily absorbable, it is more soluble and absorbable, and it gets absorbed in the stomach and duodenum. Okay, next, iron transport, excretion and functions. Transport in plasma, as I told you, after absorption, this ferrous iron is again oxidized to ferric iron by ceruloplasmin. Okay, and this ferric iron is then incorporated into specific iron binding protein, and it gets transported to bone marrow. Okay, this is how this ferric iron is readily absorbed, that is, it is readily picked up by bone marrow from blood. It goes to bone marrow because that is where hemoglobin is synthesized, that is where RBCs are synthesized. Okay, so RBC contains hemoglobin, that is why iron raw material is transported to the bone marrow. Okay, excretion, only minute amounts are excreted in urine, feces and sweat. Relatively large amounts are lost in menstrual blood, I told you. Okay, during pregnancy, iron is lost to fetus, it is given to fetus. So the growing fetus is a parasite, it will keep on taking all the nutrition from the mother, whether mother eats or not. Okay, so growing fetus is a parasite. So during pregnancy, like all other nutrients, iron is also transported to fetus, lost to fetus. Iron is also lost from skin by means of hair loss and nail clipping. We cut our nails, some amount of iron is lost in those nails also. Okay, next, functions. What are the functions of iron? Transport of oxygen to tissues in the form of hemoglobin, involved in the process of cellular respiration. It is an essential component of hemoglobin, myoglobin, cytochromes and respiratory enzymes. Non-heme iron is utilized in iron sulfur proteins of respiratory chain. These are the functions of iron. Don't get confused. These are the functions of iron. Okay, next, variations in hemoglobin concentration. Conditions that decrease hemoglobin concentration: Physiological, in children have lower value, women have lower value, okay? Physiological, they have lower value comparatively. Pathological, anemia and pathological conditions which produce hemolysis or hemodilution, okay? These will cause decreased hemoglobin. The conditions that increase hemoglobin concentration physiologically, high altitude due to hypoxia. When there is hypoxia, hemoglobin percentage increases because whatever whatever oxygen should be drawn from the air, more hemoglobin is put into work, more RBCs are put into work. See, hemoglobin increases means it is not that hemoglobin directly increases. Number of RBCs will increase. Thereby, hemoglobin also increases. It is increased in newborns. Okay, physiologically it is increased in newborns. As I told you, within 8 to 10 days there is hemolysis. In excessive sweating, there is due to hemoconcentration, there may be increased hemoglobin. And pathologically, polycythemia vera and other conditions like congestive cardiac failure, congenital heart disease, where there is hypoxia, emphysema. In such conditions where there is hypoxia, even then due to hemoconcentration, there is increased hemoglobin. Those are pathological conditions. So when there is hemodilution, there is decreased hemoglobin. When there is hemoconcentration, there is increased hemoglobin. Okay. Next, hemoglobin destruction or degradation. When RBCs are destroyed after lifespan, after RBCs complete 120 days, RBCs are destroyed. Hemoglobin is degraded into heme and globin. Again, how it forms. The wear and tear, if the protein has wear and tear, those amino acids are phagocytosed. Other amino acids which are intact are given back to amino acid pool. And heme is again split into iron and porphyrin. Iron is hemosiderin. Porphyrin is hemotoidin. So iron from heme goes back to bone marrow. Okay, for what do you say? Heme amino pool. For recycling, for using in next RBCs. And porphyrin is converted to biliverdin, which is converted to bilirubin, and this bilirubin is excreted into the small intestine through bile. Okay, bile is called bile because it has bilirubin and biliverdin. So this bilirubin is excreted into the intestines and finally excreted in feces. Some of the bilirubin is also there in blood, which is excreted through urine. Okay, so this is how hemoglobin is degraded. Next, applied physiology. So this is very important nowadays because glycosylated and glycated hemoglobin. So glucose, see, all proteins have an affinity for glucose, or rather the other way around, glucose has affinity to all proteins. Okay, same way glucose gets attached to hemoglobin also. Okay, this results in a structure called glycosylated hemoglobin. Okay, this is called HbA1c, what we check in case of diabetes. Okay, see, normally also in normal people also around 4% of glucose, around 4% of hemoglobin throughout circulation is glycosylated. Okay, that is normal. That is why when you read an HbA1c report, it shows that 4 to 5.6 is normal. Okay, so generally in people who are normal without diabetes, the glycosylated hemoglobin is less. Okay, this is very important. The fraction of hemoglobin known as glycosylated hemoglobin is clinically important. In normal individuals, it is less than 4%. When there is chronically elevated blood glucose, see, in case of diabetes, that is why it is also called as 3 months average, because chronically elevated, when blood glucose is chronically increased, it is more every day for a very long time, that of course definitely more hemoglobin gets glycosylated. Clear? So that is why the glycosylated hemoglobin percentage increases. So 5.7 to 6.5 is pre-diabetic, more than 6.5 is diabetes. Okay, that is how we will know the glycosylated hemoglobin shows that the elevated blood glucose level is chronic. It is from a very long time. That is why it is also called as 3 months average in local language, or in layman terms, in non-medical people, it is known as 3 months average, because it shows chronically elevated blood glucose. Okay, next, glycation of hemoglobin is different. Glycated hemoglobin is different. Glycation of hemoglobin is different, because here, when glucose is attached to hemoglobin by non-enzymatic process, it forms glycated hemoglobin, which is pathological. It is not normal, because glycated hemoglobin, glycation requires two factors: increased sugar and also increased free radicals. Okay, that is why glycated hemoglobin, if it is increased, it shows there is oxidative stress in the free radicals, and there is oxidative stress. Here where these antioxidants, which are rich in fruits and vegetables, where fruits and vegetables are rich in antioxidants, if you are not consuming more of fruits and vegetables and foods rich with fiber, these antioxidants, they neutralize free radicals. Okay, so if free radicals are more, it shows that there is oxidative stress. And glycated hemoglobin shows the presence of free radicals. Okay, next, anemia. We know anemia is defined as reduction in oxygen carrying capacity of blood. There are various types of anemia depending on whether hemoglobin is reduced or there is RBC defect. Okay, so overall, if the oxygen carrying capacity of blood is reduced, that is called as anemia. Okay, next, hemolytic anemia. This is destruction of RBC. Hemolysis leads to hemolytic anemia. See, when there is defective RBC, or in physiological conditions like newborn baby, there is hemolysis. So this leads to, see, in newborn babies, even if there is hemolysis, excess hemolysis, the kidneys have the, what do you say, capacity to take it away, the clear the excess hemoglobin. But if it happens in adults, it is pathological. Okay, so it can affect in two ways. It will cause hemoglobinemia and hemoglobinuria. So this hemoglobin is released into blood, causing hemoglobinemia, and this goes into renal circulation, and it is filtered in renal tubules to appear in urine to become hemoglobinuria. Okay, so hemoglobinemia will increase the blood viscosity, which will affect cardiac output and alters blood flow dynamics. Okay, and hemoglobinuria causes acute tubular necrosis, because it forms a protein complex and blocks the sieve, that is the Bowman's capsule. Okay, and that is why it causes acute tubular necrosis and acute renal failure. Okay, so hemoglobinuria is dangerous, and excessive hemolysis also causes hemolytic jaundice. Okay, this we have seen in neonatal jaundice, which is physiological, but in some pathological conditions, like where the RBCs are defect, like sickle cell anemia, okay, or microcytosis, macrocytosis, or increased reticulocyte count, in all such cases, all those RBCs, deformed RBCs, are destroyed. Okay, so in such cases, when they are destroyed in excessive amount, it may lead to hemolytic jaundice. Okay, so this is about hemoglobin and its structure, functions, iron metabolism, and applied physiology. Okay, next, these are the probable MCQs. Hemoglobin is a what kind of protein? Conjugated protein. Hemoglobin constitutes approximately what percent of dry weight? Almost 90% of dry weight of RBCs is hemoglobin. Each hemoglobin can bind up to 4 oxygen molecules. Heme consists of tetrapyrrole ring and central iron. Tetrapyrrole, 4 pyrrole molecules with 1 iron molecule. Okay, next. The enzyme that catalyzes the first step of heme synthesis is ALA synthase is the first enzyme. Okay, protoporphyrin 9 combines with which ion to form heme? It combines with ferric, Fe2 plus. Okay, next, adult hemoglobin constitutes approximately 97% of circulating hemoglobin is adult hemoglobin. HbS, that is a pathological hemoglobin. It results from substitution of valine for glutamic acid at the 6th position. It causes sickle cell anemia or sickling of RBCs when it is exposed to hypoxia. HbC is formed due to substitution of lysine for glutamic acid. Okay. Normal hemoglobin concentration in adult males is 14 to 18 grams per deciliter. Iron in food is primarily present in ferric form, not ferrous form, ferric form. That is broken down. This ferric form is broken down by vitamin C and gastric hydrochloric acid to ferrous form, which is readily absorbable, soluble and absorbable. Okay, iron is transported in plasma bound to ceruloplasmin. It is a copper-based compound, ceruloplasmin. Normal serum iron concentration is approximately 75 to 175 micrograms per deciliter of blood. Glycosylated hemoglobin reflects average blood glucose approximately for 2 to 3 months. I told you. Chronically elevated blood glucose. Okay. HbA1c more than 6.5 is diabetes. Physiological increase in hemoglobin concentration occurs in high altitude. In hemolysis, free hemoglobin in blood is called hemoglobinemia. Hemoglobinuria may lead to acute tubular necrosis. Excessive hemolysis can cause hemolytic jaundice. Hemoglobin acts as a buffer by helping maintaining blood pH. So these are the probable SAQs. So that's all for today. Okay. We'll see you tomorrow. Good night.